basically, if you've got turf, you don't have much CCA, right? That's basically the rule, okay? And it's the same, if you have lots of turf, you don't have as much coral, right? That's the thing that's going on. And we see this all through that Central Pacific. Okay? And in fact, turf is the algae that supports the greatest production of DOC and microbes. Okay? So this is an experiment that was done by Andy Haas and the Maria working group, right? And so, and was published, I guess, 2 years ago now, right? Almost. Basically, the idea is that they threw corals and algae in buckets, like one of those true marine biology things, and then we measured, incubated, and then measured, counted microbes and measured DOC over time. Okay. And it normalizes the amount of area, and then we said, well, if we have this much turf on the bottom, what would that be based on what we know the bottom types look on different coral reefs of the system? Okay? So here you have, this is the amount of DOC that would be released per by the algae in the corals, and this is the predicted microbial yield, how many bacteria we would get for that. And so if you start off with basically this idealized reef that doesn't have, it's basically only corals and CCAs, right? You get the smallest amount of microbes and the smallest amount of DOC production, right? And then if you go to this horribly thing where it's only turf algae, then what you'd end up with is the most bacterial production and the highest DOC rates. And to give you a feel of where real reefs exist, this is Kingman Reef, which is one of the healthiest ones, right? Has actually, of course, the lowest DOC release rate and the lowest predicted microbes, which is exactly what we predict or observe in that system. And Christmas, which is a degraded reef, is, of course, got higher microbes, and we assume higher DOC rates. Now remember, this is a dynamical argument. What we actually observe on Christmas, and you guys can't fuck this one up, so you got it like everybody else does, all right? What we actually observe, if you look at standing stock of DOC, is less DOC in this system, right? So basically, the coral field cannot deal with this, right? Because it's too complicated. What's going on is you release more DOC, so you chew into the refractory DOC, which is about 70 micromolar. You support more microbial production. Okay? It's labile DOC versus refractory DOC. It's not just on coral reefs, you also see it in the Arctic, and you see it in places where fresh water comes into the environment. It's probably happening all over the place, but people haven't studied it well. Yeah.
Do you have any idea how microbial diversity fits on this? Like, is it inverse, stable?
I do. Microbial diversity goes up as you get more degraded.
It goes up with an increase in DOC.
Yeah. But you also have more, that's intermediate disturbance, so I don't know, I can't really tell you, because you also, the bottom now has all these different things living on it, right, that you would have not really observed in Kingman. So even if it's just shedding off the bottom, we should have more. Yes, sir?
So this is one of the things, I was talking to Eric about this with diversity, and how there's also the evenness.
Yeah.
What do you know about the evenness of it?
Yeah, so the evenness, that's a good question, and so I don't remember quite specifically in this case. In fact, we wrote a paper that was never published. Stuart's the lead author.
Okay.
But I'm not exactly
sure why we
never published that. I think it's just sitting there on someone's computer. To go to the diversity is that I would expect in a system that is sick, you should see your evenness should change, where you have more a steeper curve, like you should have some things that are much more abundant.
So whenever someone says higher diversity,
it looks like diversity at that total diversity metric would probably look like it goes down a little bit, but richness would get higher.
Okay.
All right, so taking this data, this is going to be this is where how Tracy's stuff comes into it. Okay, so now remember. Here, these are the American protectorates by the NOAA surveys, and we go out with them and we get we do the microbial part of it. And this is just different regions. These regions don't matter so much. These are more geopolitical than anything else, all right? So we're going to go out and we're going to survey these for nutrients and stuff. And basically, this is where she brings in this MTE stuff. And I'm not going to go through that because you guys already just saw her present it, all right? So, but basically, the MTE thing is a prediction of rates at which the microbes are doing things. And so that's this, and we said the relative rates of microbes doing things versus the total rates at which things were being done on by the fish and the microbes, and that's what this microbialization score she came up with. And then if you compare this against another score, which we call the MC score, which is like a measure of human impact, you see that there's this nice correlation. And in fact, I think that the microbialization score is actually quite a bit better because things like this really stick out. So this is a reef that actually we've really fucked with out there in the ocean. Okay, so it should actually probably be much more tilted this way. Okay, and this is a reef that I have no idea why it's this high because it's actually a really nice reef that's been well taken care of. So it should be this way. And I'll bet you that the microbialization score is actually telling you instantaneously a better thing about the trajectory of the reef than any of these other like kind of combined metrics out there. Good? Okay. So this gives you a real thing. It's published, by the way, so you can just read it if you want. Okay. Now the other things from Tracy's data, which I want to make sure that everybody gets, okay, there is probably an intermediate disturbance or an intermediate microbialization state in her data set. Okay? And I haven't shown you the Dinsdale stuff, but we saw that in the Line Islands. So basically what we saw is that as you went from Kingman to Palmyra and then to Fanny, we saw this increase in the autotrophic bacteria until we get to Christmas, where everything is just completely fucked up. Okay, and as we go across that ratio, as we go across those intermediate places, we see this increase in the autotrophs. That's exactly what she saw on the whole Pacific. Okay. So there's probably this increased microbial or intermediate microbialization is associated with the rise of the turf on the benthos, and that's probably where the dam stuff is happening now. Okay, so if you're imagining this, so her stuff is the water column. This is what's happening on the bottom is the dam thing. So you can imagine that the microbial community has switched into an autotrophic thing because there's so much stuff going on the bottom here. All right, so this is what we think is going on. And then the other thing I want you to remember here is that her things actually predict that microbialization is going to get quite a bit worse when we increase sea temperature just by a little bit. Because the microbes disproportionately respond to changes in temperature. All right, good? All right, so let's go back to the interaction zones. Okay? And I want you to remember this because this is just a geometric argument of why you would kill off a coral reef. Okay? If you're a coral fighting, okay, and you're a big coral, okay, you have all of this area to dedicate to fighting on your edges. Right? And then we know that if they do photosynthesis here, you can see the stuff that was fixed here end up here, right? So it's actually they're able to recruit from that whole thing. Okay? And then as they lose space, right, they've got less and less area, right, which is decreasing with the square, right, versus a linear change. Okay? So they're still fighting the same battle lines basically, but they're losing more and more area to support it. Okay? Conversely, the algae is gaining, right? Okay? And why that's really important is that I'm not actually telling you that I think the algae is sharing stuff between it. It doesn't even matter, right? What it means is that the bigger that area gets, there's a certain point where you can't add enough grazers to it. It just doesn't matter how many grazers are there because they can only eat so much. And there's only so much habitat for them on the reef, so you never see it get grazed down.
So,
Yeah.
have you guys thought about roughly what that tipping point is?
Yeah, we've thought about it a lot, and we're starting to get there. But no, I don't have a great answer for you. Okay, but this is, I think, just a this is just simple geometry that will tell us exactly that that's what's going on. Okay? And that, of course, leads to this thing, which is what's going on with these interactions. And we've been thinking about this forever, but it's starting to become much better because there's actually more people thinking about it, which is one nice thing. The other thing that I really like are these addition of these experiments, these survey methods. Because what we're doing, what we've been doing since, I guess, the Southern Line Islands cruise, is laying down a transect tape, and then we'll go along, and what we're going to do is we're going to go to this coral, and we're going to say, well, this coral, it's this big, right? And it's interacting with CCA here, okay? And it looks like the CCA is, in this case, it would be neutral, right? So they seem to be just talking to each other, but the coral doesn't seem to be losing, the CCA doesn't seem to be losing. And then we guesstimate how much interaction is due to the CCA and then how much was due to the different algae and so forth, okay? And it's actually not so bad. It takes about an hour underwater to do maybe, let's say, 10 meters of this stuff. So it takes a long time, but it's not horrible to do, okay? And these are the things we record, and then we take those back and, oh, this is just